So thank you for the opportunity to talk about my work. I don't have a CRCNS grant, but I haven't lost hope. So instead of reading this long title, let me get into the presentation and give you a little bit of background. So we're studying a system called the vomeronasal system. It's a chemosensory system that's dedicated to processing cues from other organisms, and notably also conspecific. So it's involved also in social behaviors. Now, the system is present in most vertebrates. We don't have it, but most others do. And it exists in parallel to the main olfactory system that you've heard about in at least two talks today, and it shares many similarities with that system. So it also includes sensory neurons in the nasal cavity. They're located inside a special structure called the VNO, vomeronasal organ, and they send their axons to the accessory olfactory bulb, which is a specialized region of the entire olfactory bulb, which includes the main olfactory bulb. And there, these axons interact with mitral cells, so Kevin gave a great introduction for me. And these output cells in the accessory olfactory bulb relay all the outputs to other brain regions. Now, many stimuli that are detected by the vomeronasal system elicit behaviors that are considered innate, like defensive or reproductive or parental behaviors. And you might think that because of this, the neuronal organization of the structure may also be hardwired innate. But this is actually not the case at all. The AOB is pretty much of a mess. Here you see a histological section of it, or a drawing of the cellular structures. And for example, the one receptor, one cell, one mitral cell organization that Kevin mentioned is not present here in the accessory olfactory bulb. And instead, AOB mitral cells, they sample inputs from multiple glomeruli in a manner that seems at least to be random. And so this is just a schematic of the wiring within the AOB compared to the MOB. The details don't matter now. And one of the implications, there are other reasons for this, but overall, we believe that the chemical information about certain attributes of another individual are encoded by a combination of cells in the accessory olfactory bulb. So one mitral cell would not be enough. Several mitral cells are needed in order to do that, and that's just to say we did analysis. I'm not just saying this without any backup. Of course, you're not supposed to understand these figures from this quick description. Now, things are also, and again, it's touching Kevin's talk, things are also messy in the temporal domain. So here responses are not only very much more sluggish and slower, but there's a lot of variability in the timing of the responses relative to stimulus presentation. This is shown here for 3 different neurons, for several stimuli, and that's all you need to remember, that the responses are very variable. So the question and the motivation for this talk is, are there any processes that coordinate the activity of these multiple neurons that ultimately are needed together as an ensemble to represent information about stimuli? So let me say a few words about our experiments. We use anesthetized mice. We use multi-site electrodes, not yet neuropixels, but neuronexus probes, and we insert them into the mitral cell layer of the accessory olfactory bulb. And while we do that, we present stimuli. Usually these are urine stimuli, but we use various stimuli from other organisms. The details don't matter, and I don't have time for them anyway. And what we typically do is we look at single unit activity. So here you see responses. This is actually very much related to the previous talk, because these are responses of other conspecifics in the accessory olfactory bulb. But these are single unit responses, and now I want to talk about local field potentials. So local field potentials are extracellular signals at a slower frequency range compared to single unit activity, and they represent the summed activity around the recording site. And here I'm showing you an example. This is the same trace, but it's expanded on the bottom of one recording channel that we recorded. And the oscillatory event that I'm referring to is shown here in the gray box. And we saw these oscillations when we started to look at the data, and they were pretty intriguing, and they raised several questions. And these questions are they related in any way to the sensory stimulus given? And the other question that came up to mind is, do they have a role in coordinating the activity of the mitral cells? For both of these questions, we need to find a way to identify these events and to quantify their occurrence in a way that's as automatic and objective as possible. But this is tricky. And this is tricky because these oscillations, first of all, they vary during their duration. They change their frequency. And furthermore, they are embedded in the noise that our experiments inevitably have. So we have various sources of noise. And if we would just take a simple bandpass filter, we would not be able to distinguish these oscillations from the noise. And also, they're not always as nice and clear as they are in this specific example. But what allowed us to overcome this difficulty is the fact that we are using these multi-site electrodes, and that when we look at multiple electrodes, there is a very characteristic spread of the magnitudes or the intensities of these oscillations. So unlike noise signals, which I show you here, these oscillations, which are of interest to us, are very different in their amplitude across the different channels. And if we plot them according to their spatial positions from which they were recording, we can see, and that's after we compare it also to the histology of the AOB, that they are localized to the mitral cell layer in the AOB. And an important thing is, within a recording session, so within one insertion of the electrodes while we do the recordings, the magnitude of these oscillations is highly stereotypical. So when we have another event, it will look very similar in the spread of relative amplitudes, as it is here. And this allowed us to summarize this pattern of magnitudes just as a single value for each of the recording sites and use it as a template. Okay, so we consider this a template that represents the magnitudes of the oscillations across the sites for a given session. And using this, we derive a new signal, which we call the template match signal. So the template match signal is what we're doing. We're actually correlating each time point in our recording with this. It's basically a linear correlation that we truncate and that we raise to the power to make it a little bit more sharpened. And we obtain a value that's a measure of the similarity of the ongoing activity to the activity that occurs during the times that these clear oscillations are taking place. And this is an example of the template match signal for the same section of data that I showed you before. So the template match signal is shown by the blue line, and you can see that it's high during the time of the clear oscillation. It's very low. It's actually zero during the time of the noise artifact, and that's a good thing, of course. But the interesting thing is that it is also non-zero during periods where we don't see obvious oscillations. And so we can treat it as a continuous measure of the oscillatory network state, and we can ask how this depends, how the TMS, the template match signal, depends on the events of stimulus presentation during our experiment. And here I'm showing you an entire experiment where each green horizontal line corresponds to one stimulus presentation. You don't know what the stimuli are. And the blue is a template match signal, and you can see that it clearly follows the times of stimulus presentation. We can, of course, look at this in higher resolution, and one analysis that we did was to look whether this depends on the nature of the stimuli, the identity of the stimulus, and the answer is that indeed, 3 minutes? Indeed, it does. It depends on the magnitude, on the identity of the stimulus. So, in other words, this signal captures an aspect of the network activity, and it varies with the stimulation, and it's as if the stimulus is strong enough, it can trigger or induce the network to enter this oscillatory network state. So let's get back to the oscillations, specifically to the relationship between them and mitral cell activity. And in order to ask about the relationships, we were interested about the phase relationships. We used an approach called wavelet synchronized transform that allows us to extract ridges from the signal. So ridges are just traces in the time frequency domain. For example, here you see one such ridge along with the original signal. Okay, so this approach, it extracts various ridges from the signal. The first ridge is the one with the highest energy. And what I'm showing you here is a reconstruction using of several such oscillatory episodes with one ridge shown in green and with three ridges shown in red. The original signals are shown in black. And you can see that the reconstructions are good not just with three ridges, but also with a single ridge. Now, what is the benefit of this approach? The benefit is that if a single ridge accounts for the signal, we can use it to derive the instantaneous phase of the signal, and that's a single unambiguous phase, which we can use to associate each spike time with this phase. And now we ask, do these other phase distributions of the spikes uniform, or are they somehow modified by the LFP oscillation? The answer is they are modified by the oscillation. So here are several distributions of single units. If there was no dependence, you would see a flat histogram. You see instead histograms in green that are not flat. And the question is whether these are just some examples that I picked, because you would always get some that are significant by chance. But when we count the overall number of significant cases, we see that there's a very high abundance of significant cases. And so this tells us that Pablo tells me that I have 1 minute. So the next question is whether all the neurons share a similar preferred direction or not, direction that is phase. And this is the distribution of all the units, and you can see that there's a very clear preference for the negative phase of the LFP cycle. When we do this for shuffle data, we see a flat distribution that is not significantly different from a flat distribution. So there's a very clear preference for the negative phase of the LFP cycle. There's one interesting point here, that the phase difference, the phase locking is very loose. It's not a very strong phase locking, especially when you consider individual units. And I don't have time to show you, but when you can do a simple calculation, and you can see that when, even with very loose locking, if you have a large number of neurons, the effects sum up and can be very meaningful for a downstream neuron that reads this information. If you have many neurons, the chances to get a certain number of spikes or above is significantly higher, even if the locking is loose. And that's it. Well, it's not it. The main implication of this is that is this the presentation telling me something also? So the implication is that these oscillations could make transfer information to downstream regions more efficient by concentrating spike times within specific windows, and thus activating downstream regions more actively. There are also more fancier scenarios that emerge from our studies, but I don't have time to talk about these as well. So in short, we have periodic processes that occur within the AOB, reflected by the LFP, and they could organize the distributed activity patterns of mitral cells to effectively coordinate the transfer of information to downstream regions. And these are the acknowledgments of people who are involved with this work, and thank you very much. And
before running to coffee, we have time for a couple of questions. Yes, coffee or questions. Or you can always catch up questions during coffee. Yeah, Anand, please.
What do you think the source of these oscillations are? There within the AOB?
So yeah, we think that they are within the AOB. There's a whole part about the current source density analysis that we did, and we recorded with two different orientations of our probes, and we think that the mechanism is some interaction between the mitral and the granule cells, and there's very strong evidence that it's local because of the way that they're localized. But I don't have proof, but we have evidence for that.
Okay, I guess, first of all, thank you again, Yoram. Let me thank you, all the speakers of this session.